Hey everybody, I hope that you're doing well. Today we're going to start into our lecture on emotion, or our series of modules on emotion, depending on how you want to look at all these different video segments. This is one of my absolute favorite pieces to talk about in this entire class. And the reason for that is when it comes to neuroscience, I am a bit of a rebel. People in neuroscience do not like to talk about the idea of emotions and the brain. And there's a good reason for this. Scientists by nature are reductionist, and they want to think about things in very simple terms, and they want to be able to explain things. And emotions, by definition, are incredibly complicated and messy, and yet that they are so fundamentally important for so many mental illnesses that I have made a career out of spending my time trying to study them. And so I'm going to make some arguments for what emotions are and how we should probably look at them, and then we'll talk about some things that contribute to changes in emotion. So I really love this quote from James. This is one of the earliest things I could ever find about emotion, and a very, very important set of predictions comes out of this quote. So let me just read it for you for 1 second. And yet it is even now certain that of two things concerning the emotions, one must be true. Either separate and special centers, affected to them alone, are their brain seat, or else they correspond to processes occurring in the motor and sensory centers already assigned, or in others like them not yet mapped out. The purpose of the following pages is to show that the last alternative comes nearest to the truth, and that emotional brain processes not only resemble the ordinary sensorial brain processes, but in very truth are nothing but such processes variously combined. So for 1884, this is incredibly profound, and let's break it down. James proposes two different possibilities for the way that we should be conceptualizing emotions in the brain. Now, if you go look back at the first introductory lecture on the history of neuroscience, or if you just go read that chapter on your book and you think about what we know about the brain in 1884, this is incredibly profound that James is able to give us this important insight. He's saying either we have special centers that are the seat of emotion in the brain, or, and the second hypothesis is the one that he argues to be correct, is that emotions correspond to processes occurring in the motor and sensory centers that we've already defined or assigned, or in others like them that are not yet mapped out. So these correspond to natural brain processes that, as he says at the very end of this, are variously combined. So by some combination of all kinds of different sensory and motor processes, we come up with this thing that we perceive as an emotion. And I agree with James. This makes a lot of sense. The data support this. And that's the hypothesis that we're going to work on. That being said, there is no right answer here. This is my feeling, and I probably shouldn't even start you out by biasing with it. But I think that by the end of this lecture, that you will probably see where at least my evidence for taking this approach comes from. So I just want to show you here a list of things you don't necessarily have to memorize, but in this slide, there's a list of all of the different brain structures that we think about as being implicated in emotion. And you could just see the total number of these is striking. So an emotion, by definition, is part of just some other survival mechanism. Any of these different processes that go on in the brain are meant to help us to do one of a number of very simple things. We're either trying to seek out food, or we're trying to find the best reproductive partners, or find water, find shelter, keep ourselves safe from being attacked. And all of these would be the kinds of things that we would need these emotional mechanisms for. They help to drive this behavior. However, one of the things we're going to focus on towards the end of this semester, and this is really kind of the pivot point where we start to think of mental illnesses, and we start to talk about them through the rest of the chapters that we're going to be going through, that in some of these cases, things can go wrong. There are really three different kinds of emotional responses that we can have. The first is a behavioral response. The second is some kind of an autonomic response. And the third is a hormonal response. So let's take a close look at each of these specifically. So behavioral responses come in all kinds of different forms. And I think this is probably the easiest for each of you to recognize. There's some sort of a motor system response here that happens, and these are going to often be specific to the situation. So we can have things like approach or a seeking kind of behavior for things that are positive, and then for things that are negative, we might avoid them altogether, or much like the deer in the headlights kind of cliche response that we all think about for a deer on the road. You might engage in freezing, and the whole point of freezing is that an animal hopes that they're not going to be detected. The deer in the headlights doesn't realize that the car is not going to forget that it's there, but it's just part of a natural response. And these responses necessarily then are situational. So the response should match whatever the most appropriate life-saving or survival-based mechanism would be at that time. The second piece that we need to look at is the autonomic response. The autonomic response typically involves activation of the sympathetic nervous system. And one thing that you should know is that there's always some activation of the sympathetic nervous system. But when we especially increase this and put it into overdrive, this is part of a fight or flight kind of a response. And this typically comes along with an inhibition of the parasympathetic nervous system. So when we get an activation of the sympathetic nervous system, what you see are a number of different things that you might think of in a moment when you've been scared or something like that. We dilate, for example, the lungs, and we accelerate our heartbeat. And some other things may be those that you don't think of. For example, you might have an inhibition of digestion. You might stimulate some release of glucose via the liver. This is something we just talked about with ingestive behaviors. And this allows us to start using energy that we need. And we start to release hormones from the kidneys and all kinds of things like this. So you put all of these together, and they basically are a recipe for a fight or flight response. And of course, the parasympathetic is the exact opposite of many of these. And for the most part, we try and keep these in balance. But when we get into a situation where we need to activate the body, we put this sympathetic system into overdrive. Now, one of my favorite stories involving the sympathetic nervous system and our understanding of just a little bit of where its function comes from, or where it connects into the brain, is a set of studies that were done in cats in the early 1900s, around 1920, by a man named Philip Bard, and he was part of this movement that we talked about at the very beginning of the semester called functionalism, where people are trying to just cut out different portions of the brain or stimulate portions of the brain and determine what their functions are, either by what actions animals could no longer perform or things like this. This is very early days for our understanding of the brain. And so what Bard does is he does these two different experiments in cats where he cuts the cerebral cortex off, and his whole deal is that he's trying to understand the function of all of these cortical structures. And he finds that depending on how he does these cuts, that if he cuts out the hypothalamus, this area down here in green, that he doesn't have a response that he'll come to call sham rage. Whereas if he leaves the hypothalamus intact and makes this transection that manages to leave it, that animals start to show this response that he eventually begins to call sham rage. And this eventually leads him to the hypothesis that the hypothalamus is this center for coordinating these kind of autonomic and somatic responses. So what happened with these cats is that when they woke up, they were visibly pissed. They were just angry. And he started to find all kinds of correlates with this, is that these cats had an elevated heart rate, they had increased blood pressure, their pupils were always dilated, their hair would stand up on their haunches, getting ready to attack. And so this response was in pretty much all of these animals. So they show these physical signs of anger, and for this reason, he started calling this sham rage. And of course, all he had to do was just change this slight angle of his cut, and whether he included the hypothalamus or not would reliably predict whether or not the animals would show the sham rage, and even somewhat the degree to which the hypothalamus was preserved would correlate with the amount of sham rage. So you can see some early pictures here from studies, either from BARD or even follow-ups, and you can see these sort of angry, kind of lashing out responses, this just visible anger for no reason, and even something done a little bit later in monkeys that shows that whether or not you stimulate these areas electrically, which is what's happening in the case of the monkey here, or if you have this sort of preservation of the hypothalamus and elimination of the cortex, which we'll talk about as being equally important a little bit later, that these produce this sham rage behavior. Then the last component that we need to talk about is a hormonal component. And this can be really, really broadly defined as well. There are tons of different hormones in our body. There's probably a few that you think about naturally. But a couple of really important hormones are those that will contribute to any sort of autonomic response, right? They're ones that go along with all of these kind of sympathetic and parasympathetic responses. And the primary neurotransmitter that we think of as being a part of this process is epinephrine. And when we talk about this in terms of the brain, we might talk about norepinephrine, especially if it's produced and signals within the brain. So epinephrine and its neural form, norepinephrine, are incredibly important for this. And these are part of recruiting this larger response by the body. And of course, now we're talking about a chemical signal instead of maybe some of these electrical signals to the heart and other places. But this chemical hormonal signal helps us to get our body ready by recruiting and making, invigorating the skeletal muscles so that we can make behavioral responses, and also preparing the body for all those different changes in energy consumption. So if you remember the lecture on ingested behavior, there's ways we can shut down digestion or activate it. And I think this brings us to another really important point, which is that we shouldn't be considering these behavioral, autonomic, or hormonal responses in a nutshell. They're all part of a bigger response, and they all contribute to one another. So I started out this lecture by saying, neuroscientists don't really like the idea of emotions. And part of the reason for that is that neuroscientists really like things that they can measure. So a really important thing when we operationally define emotion, right? When we give a scientific definition of what exactly we are measuring and what we interpret that to be, is that we take a multitude of measures. So the way that we measure emotion in animal models is to look at these sort of indirect byproducts, the things that we've just been talking about. Is there a certain behavioral response that's repeated? Do we see certain actions in the sympathetic or parasympathetic nervous system? Or can we correlate any of these things with the repeated release of neurotransmitter somewhere in the brain or hormones somewhere in the body whenever we have this kind of response? So some common examples of these byproducts are things that you are incredibly familiar with. Facial expression. You've all seen some sort of montage of photos, and I would guarantee that with some degree of accuracy, if you were to look at the facial expression of another human being, you would have an incredibly accurate perception of what that human being is feeling right now. At least for most of you, you would have some kind of accurate perception. We can also look at behaviors like the freezing response that we talked about, that kind of deer in the headlights response, where the animals just stop, showing an expression of something like fear. So we can look for any number of different behavioral responses, and we can see if they reliably happen whenever we think a certain emotion is going on. And with repeated correlation and many, many studies over many years, we can begin to have a strong understanding that a certain behavior is predictive of a certain kind of emotional response. We can also look for the release of hormones or neurotransmitters, cortisol, dopamine, norepinephrine. All of these different neurotransmitters might have roles in different emotional responses. And that's kind of one of the most important pieces, is being able to differentiate these. And then lastly, we can talk about these autonomic responses. So if we have a change in heart rate or skin conductance or blood pressure or any of these other responses, those are all things that we can measure. And so the key here is taking something that seems at face value to be very difficult to quantify. How happy are you? How sad are you? And to tie them back to physiological measures as best we can. And then to make comparisons, and of course to have really good converging elements of an experiment where many different pieces of an experiment or many different individual experiments point to the same answer. And of course, having comparator groups, some sort of a control group or something like that. So these are all the keys to measuring emotion. But just because it's hard to measure doesn't mean we should ignore it, because emotion plays an incredibly important role in things like relapse for drugs of abuse. Any sort of drug addiction has a very strong emotional component, in all kinds of mood disorders. So how are we going to study these things if we try and ignore them when we look at the brain? This is the equivalent of the early days of psychology, where B.F. Skinner, and this is not his interpretation, but many others' interpretation of his work, is by just trying to understand an animal only from their behavior and ignoring what's going on in the brain and ignoring these neurotransmitters and hormonal responses and everything else, you're missing pieces of the puzzle. So the more pieces, the better a picture we have. And from that, you should take away that the more of these different kinds of measures that we have, the better off we will be. So my guide or my keys to studying emotion is that we have to recognize that these are complex processes and that they involve many different responses. And that means something that's really important is to determine responses that are highly predictive, especially ones that are unique to a certain emotion of interest or a certain kind of response that you want to measure. And then you use those features to operationally define what you're looking at. So if you feel that freezing is a measure of fear, then you should operationally define the amount of fear as the measured level of freezing. And be very transparent with it, because this gives it takes away the ability for an individual to doubt what you're saying, because you're calling something like fear a very specific kind of behavioral response. And another really important thing that I've started to learn a whole lot more about over time is that we have to validate that these responses are maintained across species. And really what I mean by this is that they translate into our true model system that we're always trying to work in, which is humans. So if you're working in humans, then you may not have to worry so much about a translational component. But there are cases where things don't translate very well from a mouse or a rat or some other animal into humans. And we should be aware of that as well. And the last and most important thing is that we discuss what we're measuring and not what we are inferring. And so what I mean by this is that if we're talking about results where an animal showed more fear than did a control or a comparator animal that we're trying to look at, that we talk about the level of freezing and not the level of fear. And this means that at each point, we bring everything back to the most important thing in any scientific study, which is the data. So there's a little bit of a rare insight into my thought process, but I think that's really important for all of you that might consider how you can become neuroscientists and also think about emotion, or even individuals that are aiming to work in a clinic. Now you can think about things from a more behavioral point of view. If you're trying to decide how accurate one of your assessments is, you can incorporate these behavioral observations where you decide if one of these measures changes, what does that mean for how you're interpreting an individual's level of fear or anxiety or something else? And these are pieces that often come into things like the DSM as well. How frequent is a behavior, and so on and so forth. So some really common measures in behavioral neuroscience: heart and breathing rate, blood pressure, skin conductance, which indicates the amount of sweat. So sweat puts water on top of the skin, which makes it easier for the skin to conduct electricity. Muscle tone and facial expression. So are muscles clenched or are they relaxed? Are facial expressions changed? There are actually some common facial expressions across mice and humans for things like disgust, or even pain. And then we can also get to things that are a little bit more chemical, like levels of stress hormones, cortisol, corticosterone. We can look for defecation in animals as a sign of fear or different motor responses. And of course, we talked about microdialysis in the methods lecture as a way to measure neurotransmitter levels. And there are even more up-to-date ways of doing this with some fluorescent sensors that have only been developed in the past maybe 2 to 5 years that are helping us to get an even better look at all of this. And now we're going to jump right into part 2 of our lecture on emotion. We're going to talk a little bit about fear and aggression. So probably one of the most famous places in the brain for fear is the amygdala, and many people consider this to be the fear center of the brain. Part of the way that it's gotten that reputation is that there have been so many studies on the amygdala and one specific kind of behavior called fear conditioning, where you look at the responses that animals have, these sort of emotional responses to things that should generate a fearful response, like a very aversive or unpleasant foot shock. But I'm here to do one very important thing, which is to break that myth. The amygdala is part of our limbic system and it responds to both positive and negative events. Although that means it does have a role in fear, that is not its only job. And that tends to be the case with almost everywhere in the brain. One of the things that we've now seen is that multiple sensory systems, like the olfactory system, feed into the amygdala, and this means that it is positioned to be a very powerful sensory integrator. It can take information from these sensory systems and put it together in a way that allows for learning about the environment, and that is incredibly important. One thing that we know is that all kinds of different aversive or negative events cause activation of the amygdala. So if you were using electrodes to record single neurons and you were to present an animal with something very unpleasant, like a noxious foot shock, then those animals would have an increase in the number of action potentials fired by amygdala neurons. And the other side of this is that if we have damage to the amygdala, then we eliminate a lot of the different behavioral or physiological responses that might go along with these unpleasant or aversive stimuli. And this tends to be in sort of an unconditioned form. And what I mean by that is that this is the pure response, not necessarily with any learning happening about something that might be unpleasant down the road. This is the initial response that can be lessened or eliminated in some cases by a lesion of the central nucleus of the amygdala. Some examples of this are lesions cause lower levels of stress hormones like the corticosterone, or lower levels of certain behaviors like freezing that animals tend to do when they are afraid. And there seems to be some sort of relationship between the size of the amygdala as a whole and just the levels of fear that people have. And this has been replicated enough to where there's probably some relationship there. Probably one of the more famous examples is if you happen to see the movie Free Solo, then they actually scan the brain of this rock climber who climbs faces with no ropes, and they find that he actually has a smaller amygdala than the average individual, and perhaps that leads to less fear or lower levels of these physiological responses and allows him to have better control when he's in these fearful situations. Flipping this around, if we activate the amygdala, then we can actually cause a response that looks very much like a fight or flight response. So you cause this activation, and depending on what the animal is doing or able to do, they will sometimes become aggressive. There are some famous studies where they put a fake stuffed rat into a box with a real rat, and then they stimulate the amygdala of a real rat, and it will attack the fake rat. Or sometimes if the animal can't figure out where this negative event is coming from, then they will freeze or do other things that show that they're afraid and that they're trying to avoid this negative stimulus, but they obviously can't because it's being delivered in a way where they can't control it. And actually, we can do the same thing in humans. So if we generate electrical stimulation, which sometimes happens, there are some seizure patients that have had electrodes in the amygdala, or I would assume that some more studies with transcranial magnetic stimulation have been done, and all of these allow us to cause human beings to have a feeling of fear just from this amygdala stimulation. Then, of course, as I said, the same thing happens in animals, and then one thing that we've seen in many of these animal studies is if you do this over and over and over again, which is kind of a model of something like a chronic stress, or maybe even you could go so far as to say a model of something like a post-traumatic stress disorder, then you can cause a lot of different physical symptoms to show up. And one of the most famous ones is that you can cause ulcers, which we all know that human beings get a lot when we have stress. And if you continue to repeatedly stimulate the amygdala of animals, you can actually cause an increase in ulcers. And now the entire point of having these systems in the brain that are involved in fear learning is to be able to protect ourselves. The idea is that we should experience some event in the environment, and that we should be able to have some kind of learning to where we now know information about that event, and it allows us, at least for these aversive events, to avoid that in the future. And not only can we learn this through direct experience, but we can also learn these things through observation. You don't need to drink rancid milk to not know that you should not drink rancid milk. You can observe the reactions of other individuals, or we often generalize. So we put a lot of pieces together in order to create this picture of all the things we should avoid in our world. We call these different learned emotional responses conditioned emotional responses. And this is after one of the first studies, and we just finished talking about learning and memory. So you all kind of get the idea here, is that we have some sort of an unconditioned stimulus, and this unconditioned stimulus is something that can clearly cause some amount of fear, and eventually we use other predictors in the environment to be able to predict that this thing is going to happen, and then both of these eventually are able to elicit the unconditioned response, which is the typical fear reaction or fight or flight reaction, or whatever it may be that we would have normally to the unconditioned stimulus alone. So the reason that the idea of a conditioned emotional response came about was actually because of a famous study that happened at Johns Hopkins. And this study, like many of the others that we're talking about in kind of early understanding of the brain and learning, happened around the 1920s. And in this experiment, which goes down in history as the reason we have this name of a conditioned emotional response, there was actually a young child, maybe even you could call him a baby, today we call little Albert, and he was in this Johns Hopkins study. And Albert's mom actually lived and worked at the hospital, and so she received $1 for her baby to participate in this crazy experiment where little Albert was allowed to play with this stuffed rat. And every time that he would go over and touch the stuffed rat, then this experimenter from Johns Hopkins would come in and ring this giant bell and make an incredibly loud noise and cause little Albert to become scared shitless, for lack of a better word. And so what they found was that they could then get it to the point where really anything, any sort of a stimulus in the environment that was similar, would cause little Albert to freak out. And he developed this very strong conditioned response to where he didn't like any kind of stuffed animals or things like that. And so this became this idea that an emotional response, crying and being upset, could be something that is learned or conditioned, and would cause kind of this evocation of this scary noise that's coming in the background, or a representation in little Albert's brain. And so this was kind of the one early, this is one of the earliest studies that taught us about our ability to make associations with fear. And probably one of the saddest parts of this story is that this study was published. Of course, Johns Hopkins, a very prestigious university, was published. And what people found out was that little Albert, they went looking for him. They went looking for sort of the famous baby that's part of this famous early emotion study. And they found out that Albert had actually died very, very young. Not as a result of this experiment, but actually through hydrocephalus that he acquired. But as we're going to talk about, there are some really extreme associations between emotional responses and the way that all kinds of different systems in our body work, like fear and the immune system. And so it's very important to consider. And nowadays, a study like this probably would not even be approved. The long-term effects of studies like this, because who knows whether or not there was something about the way this experiment was run that affected little Albert through many years of his life. So in rodents, we do things similar to this so that we can learn about fear systems, and this is something that allows us to begin to understand how parts of the brain process different phobias, or how perhaps traumatic events, such as in different forms of post-traumatic stress disorder, might develop. And one of the very common tasks that we use is something called fear conditioning, where typically animals are either put in a context that is paired with fear and given foot shocks in that context, and then if you test them in that context later on without the foot shock being delivered, they'll show fear. But if you test them somewhere else, then they won't show fear. Or sometimes we test for learning to specific kind of cues. So maybe a certain tone or a light or some other stimulus in the environment will then be paired with the foot shock. And then, of course, in the context, you show fear because you've learned something about all the visual cues in that environment. But also, if you go somewhere else and then you play the tone, then we can generalize any one of those specific cues to the fear response. We already said a few slides back that the central nucleus of the amygdala is responsible for the unconditioned fear responses that we can observe. And so this is something like a response to the foot shock itself, and we know that we need the central nucleus of the amygdala in order to show those kinds of responses. However, right next door in the lateral nucleus of the amygdala, which is very heavily connected to the central nucleus, we found that this area of the brain is highly important for conditioned emotional responses. And so what this means is that any of the cues, whether it be the context or discrete tones, specific kinds of cues that have been paired with some sort of a negative event, require the lateral nucleus of the amygdala in order to be demonstrated later on. So if we, for example, were to lesion the lateral nucleus of the amygdala, you might not see a response to the tone or to the context that have been learned over repeated pairings of the shock with those two cues. Now, as I've alluded to many times, if we continue to have a response to an emotional stimulus, then this can cause all kinds of physical ailments like the ulcers I mentioned before, or it can cause mental illness. And so there's a really big advantage for us to be able to make changes to fear memories and kind of update the system. And one of the ways that we do this is through top-down projections from the prefrontal cortex, and specifically we've linked this to the ventromedial prefrontal cortex. So this seems to be one part of the brain that is incredibly important for kind of changing these fear memories, or saying that that we need to update the associations that we've made. And what we found is that if you ask animals to do things where you ask them to switch an association, so you take for example, you take the tone, and there's one tone that used to be safe, and now you make that one dangerous, the predictor of this shock coming. And then you take the old tone and you now make it safe, and you ask animals to kind of do a switch like this. They can no longer make those kind of switches if we lesion the ventromedial prefrontal cortex. So this is important for all kinds of updating. All right, so switching gears now for a minute and thinking about aggression. One of the interesting things about aggression is that there are all kinds of different ways that we see this enacted in different species. So the way that we manifest kind of our fight response is very different. There are, of course, some commonalities that have been preserved across large groups of animals. Mammals tend to have a biting response that's very primal, and this is why occasionally you see boxers or soccer players get really angry and just decide they're going to bite somebody's ear off. In these sort of primal responses have been maintained, but many of the other responses are very species specific. The reason that we think we have these aggressive responses is fairly obvious, but I think it's worth noting is we first and foremost, we often need these to establish some sort of a social hierarchy. And we have a very tuned down version of this in kind of our modern refined society, but we still establish dominant and submissive positions based on all kinds of things that could be considered a bit aggressive, like posturing, or tones of voice, or anything you could think of, in that realm could be considered a form of an aggressive response. And the reason we need to establish that hierarchy is because we have all kinds of competition for different resources. These can be food or sex or shelter, any number of things like that. And so, of course, if you take this to the next natural steps, then we use these things to both protect ourselves and then to obtain all of these things that we need, especially food, for which there's often limited supply and a lot of competition among most animals in the world. So there are a few different areas of the brain that have been linked over and over and over again to aggressive behaviors, although these are not the only areas that have been linked to aggressive behaviors. One of these is a part of the brain called the periaqueductal gray, and then there's a very specific pathway from the hypothalamus to the periaqueductal gray, and also pathways from the amygdala to the periaqueductal gray that kind of link these fear systems and the Sham Rage systems that we already talked about a little bit. So this suggests that among many areas, the periaqueductal gray, along with the hypothalamus and the amygdala, are probably very important for these sort of defensive or aggressive responses that we use in order to compete within our environment. And one of the things that is well known about the periaqueductal gray is it actually contains a whole group of serotonin neurons. This is one of the primary sites for serotonin neurons in the brain. And this is important to know because there are actually a lot of links between serotonin and aggression. One of the things that we know is that if you destroy serotonin axons or you cause a reduction in serotonin production in some other way, then you can cause increases in aggressive behavior. And in fact, there was a huge study of monkeys in which they found that low serotonin levels in these monkeys was very highly correlated with risk-taking and aggressive behavior. And this study was one where they were examining an incredibly large colony of monkeys. And what they found was that the monkeys that had the absolute lowest levels of serotonin had the highest levels of aggression and risk-taking behavior. And one of the things that's sort of sad and fascinating all at the same time is that the monkeys that had these very, very low levels of serotonin were ones that they couldn't even follow through the entirety of the study, because these monkeys would get into fights with other monkeys that were far bigger than them or older and more experienced. And most of those monkeys didn't even make it through the study. On the other hand, if you have animals that have been bred for generation after generation after generation to be tame, then those animals tend to have higher levels of serotonin. So there's this really direct relationship. And we see the same thing in human beings. This is not a perfect relationship, but there is some sort of correlation between low levels of serotonin in humans and all kinds of crimes, especially things that fall into the category of violent or aggressive crimes like assault, arson, murder, child abuse, or just basic levels of aggression. And we know from twin studies, which is something that we look at a lot to determine whether something might be nature or nurture. And so one of the things that we've seen is that these low levels of serotonin seem to have a genetic component to them. So there's a hereditary component where these lower levels of serotonin can be passed from parents to children, or that they are the same between two twins. And this suggests that, there is in fact a genetic component here and not just some sort of nurture component. And we also know that drugs that can elevate the brain levels of serotonin, such as SSRIs or selective serotonin reuptake inhibitors, that block the reuptake of serotonin at the synapse and therefore increase the levels at the synapse can block aggression in various ways. Another thing that we know can influence aggression are hormones. And so if you take females who normally don't have incredibly high levels of testosterone, and you give them testosterone, then you can actually facilitate aggression. So you can see here, if you have multiple different groups and you give one group estradiol and another one testosterone and a third one a placebo, then this testosterone group in the middle is the one that has the highest level of aggressive behaviors, measured in this case by the frequency of fighting. And another really interesting thing that can cause this naturally, and this happens more in mice, not so often in humans, although it can happen in humans, is that if there's a case where females share a womb with many other males, and this happens all the time in mice who might have litters of 4 to 9 pups at any given time. So if you have a litter that happens to be 8 males and 1 female, then there's an inherently higher level of testosterone in the womb, and then all of a sudden you have a female that will be more aggressive. Another thing that is associated here with are low levels of progesterone. So these have also been associated with aggression. So there's a pretty strong link between aggression and hormone. Now, there seems to be something like a critical period for the effects of hormones as well. And this is particularly interesting because of what we all see in humans and many other mammals, where there are windows of time like puberty where aggression can become very, very important for establishing a place in a social hierarchy and all kinds of things like that. So it turns out if you castrate males, and which would cause them to have low levels of testosterone, and then you give injections that it matters that you have testosterone at various specific points. So if you give a placebo after birth and then give testosterone when the rat is or mouse is fully grown, then you end up with low levels of aggression. And if you just give testosterone early in life and then no injection later, you also don't really get high levels of aggression. But if you have high levels of testosterone early and also maintain these in puberty, then you kind of trigger this high aggressive response. So what this suggests is that at some point during development, a little bit early, but also with a component that happens later on, that testosterone helps to shape aggression circuitry. And another thing that's really important that we see here is that it does matter. It's not a perfect trigger, but it does matter that there's some sort of early testosterone exposure. That seems to be part of this critical period. So we need that to initially develop the circuit, and then we need a kind of a second hit, and people call things like this often 2-hit hypotheses, because you need kind of this first hit to prime the system and then the second one to activate it. So it turns out that we have these kind of critical windows where testosterone is critical early for developing a circuit and is critical later for then activating that circuit. And we need both of those hits in order to create this high aggressive situation. Another thing that, it doesn't really fit here, but it doesn't really fit anywhere among the ideas we're going to talk about, is that, of course, we need this aggression to be correctly directed. And there's some evidence that turns out to be really important for all the goals of these aggressive responses, like being able to engage in proper reproductive behavior, that one of the ways we prevent intersex aggression, especially in other mammals, maybe less so in humans nowadays, where we don't maybe use these cues as much as we used to earlier in our species, but there seem to be intersex cues that also act to inhibit aggression. So things you might think of like pheromones or some sort of smells that are associated with the opposite sex that allow us to inhibit our aggression responses when they are between opposite sexes, but there may also be ones that facilitate aggression among like sexes. For part 3 of the emotion lecture, we're going to try and put everything together, and we're going to talk a little bit about some pieces that we haven't been able to cover as easily in the other two segments. These don't necessarily fit into a perfect theme, but I'm going to call this impulse control and communicating emotions, but it's really kind of just the conclusion of the emotion section. One of the things I've been saying, but I'll say it again to remind you and to give you permission to do it, is if you've been sitting here for 2 parts of this in a row, you've probably been sitting for 40 minutes or so, get up, go take a quick walk, put this on pause. It's good for you. It's going to help with all of this time in work from home or school from home mode. So just pause for a second. Go take a 10-minute walk outside. Okay? If you haven't heard of the idea of pomodoros, I learned about them the other day when people were talking about how many pomodoros you get when trying to work with kids, but I thought they were kind of interesting. Maybe you take a look at that too. Just interesting ways to keep yourself working while trying to do all of these different tasks from home in a very different situation. So we talked just a little bit under the aggression section about kind of inhibiting emotions. And one of the things that we often consider is that the emotional centers of our brain may be the more primal centers. And one of the myths I always like to debunk when I say more primal is this idea that they're older parts of the brain. You know, there might be some evidence that certain structures in the brain have been retained for longer across species and evolution, but I'm not really certain we can call any part of the brain necessarily older. And yet more primal, okay, maybe we can go with that. The idea is, though, that the control here, as we've seen for many different parts of the brain, is segmented to where we have centers that are responsible for emotion, and then we have kind of higher order cognitive centers, we have motor centers, we have sensory centers, and all of these have to talk to each other. And so one of the things that our cortical structures do in engaging in this cognitive control is to help regulate emotions by being the bridge between all of these different structures and centers of the brain. And the structure we mentioned before was this ventromedial prefrontal cortex that's very important. But there are others too, like the orbital frontal cortex or the anterior cingulate cortex, especially one particular area of the anterior cingulate cortex called the subgenual anterior cingulate cortex. We keep going back to these famous cases in psychology, but the reason that they're famous cases is because they are amazing examples of what can happen. And one of the interesting things about Phineas Gage is that we still have a record of Phineas Gage's skull today, but things like autopsies and histology were not nearly what they were. So by having the skull, we only have a part of the picture, and there have been many attempts to model this tamping rod that went through Phineas Gage's skull. The most recent of which suggests that there was some sort of a hinging action, and it actually puts most of the damage over the ventromedial prefrontal cortex. But this, to this day, is an argument over what brain structures might have actually been damaged. One thing we do know from patients that have had damage due to stroke or traumatic brain injury or other things is that damage specifically to the ventromedial prefrontal cortex. So this is the part of the prefrontal cortex that is at the bottom and near kind of the middle of the brain. And by middle, I mean if you were to draw a line kind of at your sternum and up through your nose, that represents that middle plane. And the bottom of the ventromedial prefrontal cortex is probably just below your temples or somewhere thereabouts. So this ventromedial prefrontal cortex damage causes very specific changes, and this doesn't affect people's ability to engage in cognitive tasks. They can still do all kinds of reasoning. Phineas Gage was still fairly successful in doing all kinds of different jobs throughout the rest of his days. But one of the things that would happen is that he came to a point where people describe him as having something I think of as being like a snap temper. He would respond very quickly to any sort of insult. He would become enraged or would do things without thinking. And so this ability to control anything that you might think of as an emotional state was very dramatically affected. One of the things that we talked about with fear is that we've seen this amygdala as kind of an emotional center. And we've been talking a little bit about the prefrontal cortex And one of the things that is important to know is that the prefrontal cortex actually has connections where it can inhibit the amygdala. And of course, then you might imagine that if for some reason there's a failure of these connections, then it can lead to issues being able to control those emotional responses in the amygdala. So in fact, something that's been found as a part of all of this is that there's an interesting relationship between these two structures where the ratio of the size of the amygdala and the ventromedial prefrontal cortex can actually be a really strong predictor of emotional responses. So it's not necessarily the ventromedial prefrontal cortex itself or the amygdala itself, but for example, if you have a fairly large ventromedial prefrontal cortex and a smaller amygdala, this might suggest that you have a stronger ability to control those emotional responses via top-down mechanism, whereas if the opposite is true and there's sort of a larger amygdala with very little ventromedial prefrontal cortex, then it might be harder to control emotional responses. And one of the ways that you could put this all together is, if you think back to our sensory systems, we were saying the more brain that's dedicated to something, kind of the more resolution that brain area has. And we can apply some of these ideas to cognitive responses like emotional regulation. You can see here a really interesting example of just how important this ratio between the prefrontal cortex and amygdala can be. Now, we all know that children and all the way through adolescence tend to be fairly impulsive. They don't really understand exactly the consequence of every action that they engage in, and it turns out that we see something that matches this exact idea when we look at the brains of children and adolescents, where the volume of the amygdala tends to be larger than the volume of the prefrontal cortex in both children and in adolescence. But at some point, these begin to balance out, at least on the average. And there are still going to be adults for whom the amygdala might be larger than the prefrontal cortex, and these individuals tend to have criminal histories, and the interesting thing about those criminal histories is that they're associated with very specific types of crimes, that these tend to be kind of impulsive crimes that maybe of opportunity or things like that, but there are other areas of the brain that we've associated with very aggressive or sociopathic tendencies, and this is not a pathway that we associate with that necessarily. We also talked about aggression, and we talked about the serotonin system. And so you can imagine that a part of engaging in these sort of impulse control measures for any one of us involves being able to have all of these systems perfectly intact. And so when we have these decreases in serotonin, we talked about that we have increases in aggression and violence and all kinds of other risk-taking behaviors. And so this is also a part of the impulse control. We need these sort of modulatory systems or top-down control systems all to work together in the correct way. And when they don't, we tend to have all kinds of different problems. So one interesting connection here is that the prefrontal cortex that we've been talking so much about receives a lot of input from these same serotonin neurons, and that they seem to be involved in activating parts of the prefrontal cortex, which would mean that the prefrontal cortex would have more activity, therefore more connections and projections and more ability to control other areas. And so it seems as if there's a very strong relationship between these decreases in serotonin and these impulsive behaviors due to the fact that the prefrontal cortex is no longer getting all of the input that it needs from these serotonin neurons. And if individuals have increases in serotonin, or in many cases we will do this pharmacologically, then we see the opposite effect. We start to decrease these behaviors. So this shows us that we have this inherent correlation between the serotonin system and these top-down. The idea of communicating emotions is actually one of the even more fascinating parts of this entire chapter. And we could probably spend the entirety of the class talking about this, but we're just going to give a really brief overview of some of what we know regarding how we can communicate emotions back and forth. So one important question for us to ask is whether emotional responses are learned or whether they're innate. Darwin proposed that these were innate very early on in his writings. But what really is the evidence that we would have to support something like that? Well, one thing you could do is you could look at groups of indigenous peoples who may be isolated and separated. For instance, those individuals that are separated in very remote parts of the world and have little contact with other cultures. Well, if you were to look at people in those kind of isolated groups, what you would see is that the emotional responses are the same. And this suggests that all of the differences being raised in an isolated culture don't really change some of the emotional behaviors that we think of as being common to certain kinds of emotions. Along these same lines, facial expressions that we use are the same across cultures. So even though there may be subtle genetic differences, or people may be living in different parts of the world and there might be different traditions, the facial expressions still tend to be somewhat universal. Probably even a slightly stronger piece of evidence is that individuals that are congenitally blind, blind from birth, are able to show the same facial reactions despite being unable to use things like the mirror neurons that we've talked about or other things like that in order to specifically imitate these facial reactions. And so this suggests that these reactions are also there from birth, because a smile is exactly the same from one person to the next, irrespective of all kinds of different ways that you might be able to learn a different response. We maintain these responses. And so those are the sort of things that suggest some sort of a pre-existing program. And then lastly, nonverbal vocalizations can be incredibly important. So an easy example of this is the idea that you could be listening to somebody speak in another language and you would still have a fairly good idea of their emotional state or tone. We can often listen in and determine that somebody is angry or happy or sad, because when somebody is calm, they have a certain tone to their voice. And when somebody is really excited, they have a different tone to their voice. And this all has to do with those activations or inhibitions of the sympathetic and parasympathetic nervous system, and so on and so forth. And if we look at either fMRI or lesion studies, one of the things that we find is that there are a couple of brain areas that are important for the recognition of emotions, especially in other people. So what we've seen is that if you have some damage to the right prefrontal cortex, then this can cause problems with either recognizing emotions in others or producing emotions, whereas the left prefrontal cortex seems to be more important for if somebody else is describing an emotional response to you, describing the facial expression of somebody else, or things along those lines. In reality, these are almost two sides of the same coin, where we think that these different parts of prefrontal cortex are just involved in being able to recognize somehow the emotional responses of others. We also see responses that are important in the amygdala. And so people that have damage to the amygdala have trouble recognizing facial expressions, and being the quote-unquote fear center that it is, this tends to be a little bit biased towards things like fear. And there are also some cases where we have recordings from seizure patients where they have micro wires that are in the amygdala. And so for these seizure patients, we can record what the neurons in the amygdala are doing. And those neurons are often tuned or have the highest rate of action potential to responses that would generate fear. And this doesn't mean that there aren't neurons that respond to things that are positive or other things like that, as we've already said, but there does seem to be a strong functional role for the amygdala in fear. And then lastly, we already talked a little bit about mirror neurons, so I'm just going to bring them up very briefly here since we're talking about facial recognition and recognition of emotions. But we know that mirror neurons are very important for empathy because they cause our sensory and motor systems to get some information about the exact type of motor responses that another person is engaging in. So if we know that somebody has their face and they're making kind of a grimace of pain, then we sort of mirror that system back in our own brain, and we begin to understand that looks like what happens when we activate the same neurons that would be involved in pain or disgust or things. So this is the point in the lecture where we need to go back to James' hypothesis and kind of bring everything full circle. As a reminder, this second hypothesis we're talking about is saying that emotions correspond to processes occurring in the motor and sensory centers already assigned or in others like them not yet mapped out. So one thing I want to talk to you about is a syndrome called Mobius syndrome. And individuals that have Mobius syndrome have a form of facial paralysis, and it prevents them from making normal facial expressions. But one of the most interesting things about this is that these individuals also end up having trouble recognizing emotions in others. And what this tells us is that not having a normal sensory and motor system, not having that feedback, not having that information coming through, actually causes a deficit in emotions in and of itself. Another really great example comes from looking at the system that's involved with disgust. So we already talked about in the gustatory lecture that the insular cortex is basically the primary cortex for the gustatory system. So this is the system involved obviously in taste and processing things related to taste. But an interesting thing about the insular cortex is that it turns out it's next to the side of the head, and it makes it incredibly prone to damage. And so what happens is in individuals that have damage to the insular cortex end up with a completely normal array of emotional responses, except that they have trouble recognizing the one type of emotion that relates back to that specific sensory center, and that is disgust. So what this tells us is that in kind of a functionalist perspective, we need these sensory centers in order to be able to process emotional responses that are in fact related to that kind of a sensory, or in some cases with the Mobius syndrome, a motor response. So I think we can even do a little bit of an experiment here. And the experiment is better done in class where you could look at a partner. But what you could do is if you just take a second and you have some kind of a mirror nearby. So you could pause for a second and move your computer over near a mirror. You could look, or you can even take this list if you want, and you have a member of your household that you want to try playing this trick on. Then we'll go through this experiment. So what I want you to do for this experiment is if you're doing it on another individual, you can ask them to close their eyes and you can write down the list, and then you can go ask them to just think about everything that you're saying. And if you're looking in the mirror, obviously you have to look at yourself. And in that case, just see if I get you on one of these. So what we've probably all had something that we think of as kind of a conditioned taste experience. For many people around the Rutgers area, this might involve some sort of experience with alcohol at a bar. That tends to be a really strong one. And so when you tell people to think about tequila or Captain Morgan or any number of other alcoholic drinks, sometimes that's a really good one for getting a response. But try this list on for size. So take a second, clear your mind, and just listen. Spinach. Biting a lemon. Brussels sprouts. A shot of tequila. Liver and onions. Chocolate. Cottage cheese. Ice cream. Mint. Vomit. So if you did this experiment the right way, then you probably ended up with one of these responses. This is, of course, kind of that universal look of disgust. And I'm certain that if you got that out of yourself or your brother or your sister or whatever parent you decided to run this list by, you didn't have to ask them what the face that they were making was, because you already knew in the back of your head what that look of disgust appears like on so many other individuals in the world. And this is kind of one of the the main features that we're talking about here. These responses are universal, and that's all that we really need in order to understand how somebody else is feeling about a certain kind of food or something like that. We recognize disgust when we see it on others. I'll be the first to say that the book spends most of its time talking about negative emotions, but we'll spend a little bit of time here talking about positive emotions. So one of the areas that's been repeatedly implicated in happiness, and I think that for me personally, somebody who studies reward motivation, that a lot of these are broad overgeneralizations, but the right prefrontal cortex seems to be more involved in happiness than the left, even though both play a role. And for many of us, probably due to handedness and things like this, smiles are more intense on the left side of the face, which is controlled by our right hemisphere. And so this is something that we see across many humans when their prefrontal cortex happens to be recorded. Just like we saw in the case of Phineas Gage, if we have lesions of the right hemisphere of the prefrontal cortex or in the left hemisphere, it doesn't really matter. We saw examples of both a little while ago for other emotions, but we can have different effects on emotional responses. For example, lesions of the right prefrontal cortex can affect the emotional tone of vocal expressions, which is something we were talking about. And then we did talk a little bit about the basal ganglia as being important for kind of motivated behaviors. And we know that the basal ganglia plays a really important role in positive emotional states and processing things that are valuable or that make us feel good or that are rewarding. And in cases where we have some sort of dysfunction of the basal ganglia, we talked about a couple of examples. You can also have corresponding changes in emotions. Okay, so let's start moving in the direction of wrapping this up here. I think this diagram that I've got up on the screen does the perfect job of going through everything that we've been talking about. When the brain produces a reaction, then we engage multiple different responses. We have a behavioral response that comes from our muscles. We have the engagement of the autonomic nervous system that creates all of these different autonomic responses, typically in the sympathetic or parasympathetic nervous system. And then we have the hormonal or endocrine responses. And for each of these, this is all going to provide feedback. So when we start to engage in a behavior, we know that we have signals that are coming back up into the brain. And the same thing with these autonomic responses. If the heart starts to beat faster, we've already talked a little bit about having a system. We talked about this in relation to thirst, but it doesn't really matter what it's in relation to. We already have a system for detecting blood pressure. So when we start to physically increase our own blood pressure, we have a feedback system. And the same thing that with the endocrine system. These are chemicals that are then being secreted, and then we have receptors for those same chemicals. And all of these things necessarily feed right back into the brain. So much like we saw when we looked at hunger and thirst, emotions become a closed loop system. This example of the muscle here is probably the easiest. You get angry, you clench your fist. And then we've already talked about the fact that we have all of these intrafusal fibers that are kind of sensory and muscle fibers that are responding to how much the specific muscle that we're engaging is stretched or flexed. They respond to different levels of extension or flexion of that muscle. So immediately when we start a behavioral response and you clench your fist in anger, you say, I'm so mad right now, then you have feedback coming in. So let's finally take James's idea and let's look at how that could be accomplished in a physiological system. And in some ways, I know from the beginning, this is a totally reductionist way of looking at something incredibly complicated like emotions. And yet it's also a really great way of explaining how these interactions between the brain and body, many of which are circular, as we just saw in the last slide, are able to produce one of probably the more complicated behaviors that any of us can think about. So if you take, for example, something as simple as a fear response, then what we have is a different combinatorial kind of response for fear than we might have for something similar. So in the case of fear, we know that heart rate goes up, but skin temperature goes down. And so this is just, in only looking at two different physiological responses, we can see that if we look at fear and we compare it to something like anger, where the heart rate goes up and the skin temperature goes up, then now we can take two things, both of which could be considered, I guess, negative emotions in some way, but they're differentiated by at least one important feature. And in this case, it's skin temperature, the difference in the way they respond to that. And for happiness, we see something very different, kind of an opposing response, right? Because we are going to have a different kind of an autonomic reaction. So in happiness, the heart rate's going to go down. And in the case of happiness, we have no change in skin temperature. So if you then take this idea and you just kind of carry it out to the depths of infinity, where you were to take all of the different responses that we were to have mentioned, measures in the blood and pupil dilation, digestion, all of these different possible reactions the body can take, and you put them into a chart like this, and then you were to quantify emotions, then you could see that the exact and sort of precise combination of features that you would get could, at least one could argue that they could define all of the different emotional responses, right? So it's that unique set of bodily features that creates the emotion that we're talking about. Now, one of the ideas that we've been putting forth here is that we need this interaction between brain and body, and then body and brain, to complete this emotional circuit. And there are a lot of examples of this. One example is that patients, sometimes they get Botox, where you have some paralysis of the face. They begin to show things like those Mobius syndrome patients. They no longer have the exact same facial reactions as before. So what I want to leave you with are a couple of readings from a guy named Homan, who at some point in his life had experienced pre and post spinal cord damage emotions. And so he leaves some really profound writings on what those changes felt like. And I think that they demonstrate this idea, and they demonstrate some of what we've been talking about with James' second hypothesis, that we have centers that already exist and we're just changing the way that we perceive what's coming back from all those different sensory and motor centers of the brain. So Homan says, I sit around and build things up in my mind, and I worry a lot, but it's not much but the power of thought. I was at home alone in bed one day, and I dropped a cigarette where I couldn't reach it. I finally managed to scrounge around and put it out. I could have burned up right there. But the funny thing is, I didn't get all shook up about it. I just didn't feel afraid at all, like you would suppose. In another writing, Homan says, now, I don't get a feeling of physical animation. It's a sort of cold anger. Sometimes I act angry when I see some injustice. I yell and I cuss and I raise hell, because if you don't do it sometimes, I've learned that people will take advantage of you. But it doesn't have the heat that it used to. It's a mental kind of anger.